Hello, everyone. Welcome to the second day of the DeepMath conference. It's my pleasure this morning to kick off the day with Surya Ganguli. He's done an impressive range of work, sort of from domains in physics and in neuroscience and, of course, in deep learning. And he's going to talk to us today about deep learning theory from generalization to the brain.
Thanks. So it's great to be here, and congratulations to the organizers for organizing a wonderful workshop. I'm here to bring it down a level, but it'll recover hopefully afterwards. So we've actually been working on theories of deep learning, and I think there's lots of room for really interesting work combining mathematics, physics, statistics, computation, and so forth to understand deep learning. Here's kind of some of the works we've been working on over the last few years. What's interesting is we can use interesting ideas from other fields like non-equilibrium stat mech, Riemannian geometry, random matrix theory, free probability, as Jeff talked about, to shed some light on deep learning systems. I can't, of course, talk about all of this stuff, but we actually wrote a review with my colleagues at Google, kind of trying to summarize all of this stuff. This should appear in annual reviews of condensed matter physics quite soon, and we'll put it up in the archive soon. But we tried to give a coherent overview of physics-based approaches to understanding deep learning, going from expressivity, error landscape, signal propagation initialization, generalization, and generative modeling. So if you're interested, you can send me an email if you want a copy. Okay, so for this talk, I just want to give you kind of 4 vignettes, right? So first is generalization. How can neural networks predict the response to new examples? And can we understand it analytically, at least in simple settings? Then, of course, interpretability is at the core of what we want to do, both in applications of deep learning to engineering or, say, medical, healthcare, or law. We definitely want interpretability in our models, but also for scientific purposes, to use deep learning systems as models of the brain. We would like at least those systems to be as interpretable as possible. So I'll talk about obtaining interpretability through model reduction with applications of the retina. And one of the other things we'd like to understand is why do models, say convolutional neural networks, choose the particular channel filters that they choose? And we'll try to explain that also by trying to actually explain why the primate retina chooses the convolutional filter channels that it does choose. And we'll show that Sorry, that sounded like me. It's like I couldn't tell if I was saying that or not. Anyways, but yeah, where was I? Yeah, so we'll show that optimal convolutional autoencoders actually match the primate retina, and we can come up with theories for analytically computing such optimal convolutional filters. And then the final thing is explaining hexagonal lattices in the brain. These grid cells, we can show that recurrent neural networks trained to path integrate automatically develop grid cells, and we have a theory for why they do that. Okay. So starting off with probably what's most central to this particular topic of the conference, the learning dynamics of training and testing error. So and by the way, everything I'm going to talk about is actually published except the very last part, and these are the references. Okay, so this is joint work with Andrew Sachs and Andrew Lampinen and Jay McClelland at Stanford, and it's this set of papers. So here's the basic idea, right? Whenever we're doing deep learning, we're always doing optimization on the wrong landscape, right? We're optimizing on the training error, right? But we'd really like to minimize the test error, right? So even though the training error might decrease monotonically, the test error over training time might exhibit non-monotonic behavior, and that's a signature of overfitting. Of course, this learning dynamics in deep nonlinear networks is quite complex, but it turns out all of this phenomena also show up in the learning dynamics of deep linear networks. And we can actually understand these phenomena there very precisely. So what we did was we analyzed the dynamics of a deep linear network. Here I've just shown 2 weight layers, but all of our results generalized to multiple weight layers. And so this is the deep network. It transforms inputs into outputs. Okay. Because the input-output map of this network is linear, the learning dynamics is driven only by the second-order statistics of the data. In particular, the input-output correlation matrix, for example, how correlated is this input neuron with this output neuron across the training set? Okay, that input-output correlation matrix is this matrix right here. Of course, the input correlations also play a role, but we're going to assume that they're white to simplify things. We're going to analyze gradient flow dynamics, and this dynamics is a nonlinear dynamics because the loss landscape, even for the deep linear network, is non-convex. It's quartic in the weights, so you take a gradient and you get ordinary differential equations that are cubic in the weights. Okay, so it's a nonlinear set of equations, and that's where a lot of the richness lies. And we can find exact solutions. So these are old exact solutions that we found in 2014. You can understand these solutions as follows. You can take the product of the weights from layer 1 to 2 and 2 to 3, and that obeys a dynamical equation for a specific class of initial conditions. What it does is it builds up in a time-dependent fashion the singular value decomposition of the input-output correlation matrix here, sigma 3, 1, when this is white. Okay, so these are the singular vectors of sigma 3, 1, the input-output correlation matrix, and these are time-dependent singular values of this product matrix. These time-dependent singular values, if you start from small initial conditions, arise from a small value, and they rise asymptotically to the actual singular value of the input-output correlation matrix. But the time dependence of the rise is kind of interesting. It's a sigmoidal rise. So for example, larger singular values get learned earlier, and then smaller singular values get learned later, and so on and so forth. The time scale of learning of each mode, of each singular mode, is order 1 over s, where s is the strength of that singular value. So at a larger level, the take-home message is stronger statistical structure gets learned earlier, but in this simple linear setting, we know how to quantify both aspects of that statement. The strength of statistical structure is quantified by the singular value in the input-output correlation matrix, and the time scale is 1 over that singular value. Okay? Yeah, so these exact solutions are assuming that the singular vectors of the product matrix equal the singular vectors of the original matrix at initialization. So that's a special class of initializations that allows us to decouple the problem and find exact solutions. But it turns out if you start from small random initial conditions, this sort of special manifold of trajectories is an attracting manifold, and you rapidly go to that manifold and then follow it. So for example, these blue curves are what we compute from our exact analytic solutions, and these red curves are what you get from actual random initial conditions. And so they're close to each other.
But the thing is, you have to start near the origin.
Actually, you don't have to start near the origin. You can, as long as you have random initial conditions so that there's no special structure at initialization, the scale of that randomness could be whatever you want. So if you start at larger variance, you'll start up here and you'll come down. I just haven't shown those solutions here. Okay. It turns out those hurt generalization, as we'll see next, if you start with large initial weights. Okay, all of this can be generalized to deeper networks, and there's a different kind of dynamics, but again, it's sigmoidal but sharper. Okay. This is, of course, related to what Sanjeev talked about yesterday, when he was also analyzing the learning dynamics of deep linear networks. Okay, so now, in order to understand generalization, we can sort of view this entire learning process as kind of a singular mode detection wave that sweeps in from large singular values and learns the largest ones first and the smaller ones later. So for example, this is the learning, so let's say S hat is a singular value in the training data, in the input-output correlation matrix, and S of t comma S hat is the learning dynamics of the students of the network's singular value. So this is a sigmoidal rise that we've been seeing. But we can also ask, what is the ratio of the singular value of the student network divided by the singular value of the teacher, of the training data, right, at an instant of time, right, as a function of the singular value of the student, right? So then you see this wave propagating through, right? So at early times, only the large singular values in the training set have been learned, but at later times, this wave propagates inwards to smaller and smaller singular values in the training set, and then they get learned, right? So this kind of singular mode detection wave sweeping in from large to small singular values will help you in a very visceral way understand the dynamics of generalization in this simple system. So basically, at any given time t, data singular modes that are larger than t over tau, where tau is the time scale of the ODE dynamics, these are learned, but anything smaller than that time are not yet learned. Okay, so that's the basic picture. Okay. So now let's look at generalization, okay? So everything I've said up to now has not made any assumptions about the training data other than the fact that there exists an input-output correlation matrix for the training data. But to assess generalization, I need to specify a distribution over the data, right? So we're gonna follow the student-teacher framework, where we're gonna assume that the data is generated by a teacher, where you pass in random inputs, you get out outputs, but they're corrupted by additive noise. Okay, so it's a noisy teacher. We're going to work in the usual high-dimensional statistics limit, where the number of neurons in the input and the number of neurons in the output are both tending to infinity, while their ratio is order 1. And we're going to assume a certain notion of simplicity in the teacher, which is namely that the teacher has low rank, a rank that's order 1. Okay. All right, the student, we're just going to over-parameterize it. It could have many, many more hidden units than the teacher, but it has a similar architecture. Okay, we're not going to restrict ourselves to students that are 2 layers. It could be more layers, and we can get analytic solutions for that as well. Okay, so the basic idea, again, is that we know that the only thing that matters is the input-output correlation matrix of data generated by the teacher network. That's what matters for driving the dynamics of learning in the student. So what is that input-output correlation matrix? Well, it depends on the teacher only through the composition of these weights, and this is the training data. So when you actually compute the correlation matrix between inputs and outputs of the training data, what you find is that the input-output correlation matrix is nothing other than the composite teacher weights plus the noise that corrupts the outputs. Okay? So what's interesting is that the training data has an interesting low rank plus noise structure in its input-output correlation matrix. Okay? So we can exploit known results about random matrix theory about how low rank matrices get perturbed by noise to compute generalization error or test error. Okay? So there's the basic idea. So this is how the teacher is buried in the training data. And then we just put this together with how the training data drives the dynamics of learning in the student. Okay? So again, we need to understand the singular vectors and singular values of this input-output correlation matrix and their relation to the singular vectors and singular values of the teacher matrix in order to compute the test error. So again, we have these 2 matrices. Think of this as the pristine teacher matrix, whose singular values and singular vectors have the bars over them. And think of this as the corrupted data matrix, whose singular vectors and singular values have the hats over them. All right? And so the question, of course, is what's the relationship between U hat and U bar and S hat and S bar? Well, this goes back to some random matrix results by these guys, and it goes even further back to famous results like the BBP phase transition. But what happens is in this system, there exists a phase transition as a function of, say, the size of the signal or singular value in the teacher for, say, some fixed level of noise. So what happens is, let's say there's some fixed level of variance in this noise, and let's scan the overlap between, say, U hat and U bar as a function of the strength of the signal hiding inside the teacher, so as a function of S bar. So what happens is if S bar is small, there's no overlap between U hat and U bar. The singular vectors of the noise corrupted matrix have nothing to do with the singular vectors of the teacher matrix. However, as the strength of the teacher signal gets larger, there's a phase transition where the overlap starts to acquire some non-zero value. And of course, for very large singular values, the noise can't knock the singular vector off that much, so you get a pretty large overlap. So basically, there's a phase transition from which U hat and U bar are perpendicular over here, but then they acquire some overlap over here. Okay? And similarly with S hat and S bar. So in the region where there is no overlap, what happens is you almost can't even see the teacher buried in the training data. And so then the training data is just noise, so it has a Marcinko-Pasteur spectrum. But as the singular values of the teacher become larger, they eventually pop out of the noise, and they grow as the singular values of the teacher grow. And this is what's going on here. So S hat is flat because it's stuck at the edge, or maybe even within the edge. And then as S bar grows, S hat approaches S bar. Okay? So clearly, the signal-to-noise ratio is playing a strong role. And we expect at large signal-to-noise ratios, sigma 3,1 will approach W bar. And then the learning dynamics of the student will approach W bar, and generalization error will be small. But of course, the question is, what happens in intermediate regimes? Okay, so now, this is, so you can put together the random matrix theory with the learning dynamics I showed you and get analytic formulas for both the training error and the test error as a function of training time. And this is what you find. This is both theory and numerics, and they match for a rank 1 teacher, right? So imagine that there's only one outlier eigenvalue, singular value here. So what happens is the training error suddenly drops, and then it plateaus for a while, and then it slowly drops down again. Why is that happening? Well, now we can think about this singular mode detection wave, right? It's coming in from positive infinity, and as soon as it crosses one of the outlier teacher singular values, it'll learn something about the teacher, and also learn something about the training data, and therefore the training error will drop, okay? Then nothing happens for a while because the singular mode detection wave has to travel from this singular mode to this Marcinko-Pasteur C. And then once it enters the C, it learns about the noise in the training data, and then the training error drops. But the test error does something very different. When the detection wave passes an outlier singular value, the test error drops because it learned about the teacher because the singular vectors of the training data are related to the singular vectors of the teacher. But then, as it enters the Marcinko-Pasteur C, the test error rises. Because, again, you're learning about this noise, and the singular vectors associated with this noise have nothing to do with the teacher. They only have to do with the training data. Okay, so that kind of explains the shapes of these curves, okay? So again, this was a, oh, yeah?
This is where there's just one singular value? Like, when you expect to drop for each one?
Yeah, so that's what I meant. This is a rank N student and a rank 1 teacher. If you had a rank 3 teacher, you'd see 3 drops, exactly, yeah. Okay, now here's what's interesting, okay? We again have analytic formulas for the test error at this optimal early stopping time. And it turns out these analytic formulas are completely independent of the number of hidden units in the student network, as long as the student network is expressive enough to mimic the teacher network, i.e. the rank of the student is bigger than the rank of the teacher. Okay? So what does it depend on? Of course, it depends on the singular values of the teacher. It also depends on the initialization of the student at time 0. The smaller the initialization, the better. Because, of course, the initialization is random and it can't know anything about the teacher. And if you're regularizing by stopping at an optimal early stopping time, you never forget your initialization. So therefore, making your initialization as small as possible helps with test error. Of course, you'll run into optimization issues due to vanishing gradients. It will slow down the training. But if you're willing to wait, you always do better with a smaller initialization because optimal early stopping is essential for getting good test error here. Okay, so the fact that it doesn't depend on the architecture of the teacher, of course, suggests that if we're to search for any upper bounds on test error that involves some measure of the complexity of the student architecture, those upper bounds will necessarily not be tight, even in this simple setting. Okay? So as Sanjeev, for example, was saying, the entire trajectory matters. Where you start initialization, when you stop, and so forth. Okay. Here's an example where the theory generalizes to multiple hidden layers in the student. So here's 5 layers of neurons, 3 layers of weights, a rank N student, a rank 1 teacher. Things just become even sharper at deeper networks because of the cooperativity and the learning dynamics that's introduced by multiplying the weights together. Again, the test error at the optimal early stopping time is independent of the number of student hidden units. It only depends on the structure of the data and on the student architecture. Oh, by the way, that's another important lesson. Any data-independent upper bounds on test error are likely to be incredibly loose in this setting because the structure of the data plays an essential role in the analytic formula for the test error at the optimal early stopping time. Okay. All right, so does all of this break down if you go to nonlinear networks? Of course, we don't have an analytic theory for the learning dynamics of nonlinear networks, but we kept everything the same between this simulation and this simulation. But all we did was we added leaky ReLUs at all the hidden layers. And you see kind of a qualitatively similar picture where there's a sudden drop in training and test errors, and then a slow drop in the test error, and a rise in the, a slow drop in the training error and a rise in the test error. So the qualitative structure that we can understand analytically in the linear setting sort of persists in the nonlinear setting here, yeah. Oh yeah, so then what would happen is the training error would drop slowly and immediately. Well, as soon as it hits the Marshall-Capacitor C, and it depends on your initialization. If your initialization is small, it depends on various things: the scale of the teacher, the scale of initialization, and the scale of the noise. But in a certain setting where the teacher has a large scale and initialization is small, the test error will just rise. Yeah. Okay. So you'll never learn anything about the student. About the teacher, because it's so buried in the noise. Okay. Okay, so of course this is all, a lot of this work was motivated, of course, by this paper on rethinking generalization. And there was an interesting observation in that paper. So this was the paper where they showed that these deep networks have very high learning capacity because they can also memorize completely random labels, right? Yet they still generalize well on real-world data sets where the labels are intact. However, there was a difference between the learning dynamics on random data versus structured data. And the main difference was that the learning dynamics was slower. So this is data from this paper, complicated nonlinear networks, convolutional networks, things like that. So if you completely have random labels, the learning dynamics is much slower. But if you have true labels, the learning drops immediately. Okay? Okay, so we can, of course, reproduce that and understand this even in the simple linear setting. And the basic idea is, if you take the input-output correlation matrix where you have, say, outlier structure, i.e. the teacher is not buried in the noise completely, and then let's say you randomize the outputs while preserving the output variance. So you destroy correlations between inputs and outputs. Then what you do is you spread out the noise a little bit, but you destroy, of course, the outlier singular values. So in this setting, the singular mode detection wave will cover these outlier singular values early on, and so the learning will be fast. But here it'll take longer to get to the Marcinko-Pasteur C in the random data set, so the learning will be slow. Okay? So that kind of pops out simply. If you destroy strong statistical structure in your data, learning should be slower. Hopefully that's a generalizable take-home message. Okay. But here, of course, we can understand it analytically. All right. So again, just a take-home picture intuitive take-home picture is that, of course, in machine learning, we're always optimizing on the wrong landscape. We're optimizing on the training error landscape. And so we may not want to get to the bottom of that landscape. We may want to stop early to have a hope of getting to the bottom of the test error landscape, which we never have direct access to. Okay? All right. So again, just to summarize, we analytically computed training and test error as a function of training time in deep linear networks. The results qualitatively reflect, at least in our hands, learning in deep nonlinear nets. The test error as a function of time depends in a sensitive but completely computable way on initial student strength, on the teacher rank, on the teacher SNRs, but it doesn't depend on the student network size. And these networks, what happens is they seem to learn the most important structure in the data first and then only learn less important structure. So the trajectory of learning matters. It explains why learning scrambled data takes longer than learning structured data. Any attempt to bound test error in terms of, or generalization in terms of student network architecture or capacity alone is likely to yield a loose bound. So data matters. I think one of the major impediments to theory in deep learning is that we don't have good models of the data. I know the work that Haim is doing on looking at data manifolds is a great step in that direction. So to understand generalization, we need a theory of the structure in the data and its impact on learning dynamics. And it quite possibly might be that the generalization success of deep learning originates through a conspiracy between data structure and the learning dynamics of the architectures that we chose, such that that learning dynamics learns the most important structure in data earlier. That's a conjecture, of course, to be proven in more sophisticated models.
Surya,
Yeah?
there is a clear connection with implicit regularization.
Yes, yes,
and gradient descent.
yes. Yeah, exactly. So what we've,
The iteration number is
yeah,
proportional to 1 over lambda.
yeah, yeah. So here what we've,
Some nice results about.
so your lambda is the coefficient of an L2, is that?
I'm sorry?
Your lambda is the coefficient of an L2, is that what you're talking
No,
about?
no, no. So there is a almost exact connection between the number of iteration in gradient descent
Yeah.
and an equivalent regularization term. And the lambda in the equivalent regularization.
But sorry, what is that regularization term? Is it an L2 regularization on the weights or?
It's L2 regularization.
Okay. Yeah, I think that these deep neural networks do something stronger. They do a low rank regularization, right? So that comes about, I'm gonna have to go all the way back here, but that comes about from here, where if you stop early, you've only learned the largest singular modes first.
That's exactly what comes out from this analysis.
I see. So the L2 is sufficient to also induce a low rank.
2007, 2015 by a number of people, including Lorenzo Rosazzi.
Okay, so let's talk about that offline. Yeah, yeah, I'd love to understand how L2 alone can get you low rank on the product of weights. Okay, so okay, now we also we can apply these ideas actually to something that is as complicated as semantic cognition, or at least some subset of semantic cognition. So Jay McClelland, my colleague at Stanford, wrote this amazing book called Semantic Cognition. So what is this field? It's all about our ability to recognize and comprehend facts about the world, especially facts about things that we've never, that aren't currently in our sensory input. So for example, do birds have wings, right? We all know that birds have wings, despite the fact that there is no bird in the room right now. So our ability to produce semantic cognition depends on our ability to form concepts and categories and attach facts to those categories. And so there's an interesting dynamics for the evolution of semantic cognition, which is that infants tend to learn coarse-grained categories first, and then they learn fine-grained categories later. So for example, early on they can discriminate between animals and plants, and then later on different types of animals and different types of plants, and even finer scale discriminations later. Also, there's a notion of category coherence. Some categories are more coherent than others. I wasn't going to go deep into this, but I just wanted to advertise a result. So Jay actually tried to understand the semantic cognition phenomena, especially the progressive differentiation of concepts, by doing simulations back during the first time when deep networks were popular, during the PDP revolution. So what he did was he asked a network to solve a bunch of semantic tasks. So for example, here's a domain of living things, and here's a bunch of questions that you can ask. For example, does a canary or can a canary grow or move or fly or sing? And he just trained the network to answer these semantic questions using backpropagation. And he looked at the evolution of internal representations of these items over training time. Okay. And what he found was that, so this is a multidimensional scaling plot of the internal representations of these 8 items in this layer. Okay. And what he finds is from at a random initialization, they're all near the origin. But what he finds is that early on, the animals separate from the plants. Then later on, the fish separate from the birds, and the trees separate from the flowers. And then even later on, individual fish separate and individual birds separate and so forth. Right? So there is indeed this hierarchical unfolding of structure that mimics the hierarchical structure of the data set. Okay, so when I first saw that, and this is also what happens in human babies, okay? That they can discover this through looking time studies and so forth. So when I first saw this, I was at once astounded because deep neural networks could behave like babies, but I was also disturbed. There must be very nice mathematical principles governing why this happens. And so actually using the same mathematics that I talked about for generalization, we were able to recapitulate this phenomenon in a linear network and actually understand it. And basically what happens is the take-home message is, in hierarchically structured data, the input-output correlation matrix has a hierarchical structure in the singular modes, such that the singular modes with large singular value, the associated singular vectors discriminate or the coarsest categorical distinctions, and the singular modes with smaller singular value make finer scale discriminations. So this is ultimately a multidimensional scaling plot of analytic formulas that we get in a deep linear network exposed over training time to hierarchically structured data. And you can see that it qualitatively recapitulates what's going on in this much more complex data set. And so again, if you're interested in all of this, we just published a paper in PNAS just now where we looked at the hierarchical differentiation concepts, but we were also able to analytically explain a range of phenomena that occur in infant learning, including illusory correlations. For example, infants will reliably say that a worm has bones at a certain age, despite the fact that they've never seen any worm with bones. This overgeneralization that infants do early on in learning, we can explain. Category coherence, we can explain, and so forth. So anyway, all of that is here. But let's move on to the next part. Okay. Are there any questions on this part? There'll be time at the end. Okay. Okay. So now, something that I think we also need a lot of work on is interpretability in deep learning, especially in applications of deep learning for scientific purposes, but of course for societal purposes like law and medicine and so forth. Okay, so I'm going to talk about some work that we've done in the retina, where we actually have a lot of prior information about how the retina works. So we can really benchmark deep learning models in the retina, like are they doing the right thing there, okay? So here's some questions for the intersection of deep learning and neuroscience per se, and of course theory and deep learning as well. So we've obtained a lot of success in recent years as a community in training neural networks to solve a task or to mimic a data set, and then compare their internal representations to the brain. And their internal representations do indeed look like actual biological neural circuits that are also trained to solve the same task. Okay, so you solve the task, the innards of the deep network look like the innards of the brain. Okay. However, this raises profound questions about the very nature of explanation in neuroscience, right? Are we simply replacing something we don't understand, i.e. the brain, with something else that we don't understand, i.e. a deep network? Like, have we made any progress whatsoever? I mean, we've made progress in terms of explaining fractions of variance in neural data, but as my colleague Daniel Fisher at Stanford likes to say, fraction of variance explained is not the same thing as scientific explanation. There's something more ethereal, something more conceptual that we're after, I think. Okay, but moreover, beyond just getting the representations right, i.e. getting the input-output map of a biological neural circuit right, are the intermediate steps that deep neural networks take, i.e. the intermediate computational steps, to generate these representations the same in the model as they are in the brain, right? As a veridical model of a brain circuit, we want more than getting the input-output map right. We want to get the intermediate steps right. Okay? And moreover, how do we extract from these complex models mechanistic hypotheses that can indeed guide experiments in neuroscience that probe interior structure and function in biological circuits? Okay. So we've been working on these questions in a series of papers with a talented group of grad students and postdocs. So Lane McIntosh, Niru Maheswaranathan, Aran Naebi, and Hidenori Tanaka. And so there's a series of papers here. This is going to appear at NeurIPS this year. Okay, it's the last paper that I'm kind of going to focus on. Okay. So what is the retina? Okay, so the retina is actually a one hidden layer neural network at a coarse-grained approximation. So here are the photoreceptors. Photons, they absorb photons and they convert them to electrical signals. They propagate through a layer of bipolar cells, and there's lateral connectivity through horizontal cells and amacrine cells. And then the electrical activity travels to the ganglion cells, and each of you have about 1 million optic nerve fibers that come out of your ganglion cells, and those 1 million nerve fibers tell you everything you've ever experienced about the world, at least visually, right? Okay, so what are kind of the canonical models of this complicated circuitry? Well, okay, so first of all, experimentally, it's easy to shine light on the retina. It's easy to record from the ganglion cells, but it's hard to record from the interior. Okay? So the canonical model of this system, of a single ganglion cell, is that it has a linear spatiotemporal filter and then a rectifying nonlinearity. Of course, this is not really a retina, of course, because it doesn't take into account all of this complicated circuitry. How well does this simple model do? Oh, sorry. It turns out that this model actually correctly gets the retinal response to white noise stimuli at low contrast pretty well, but it doesn't accurately get the response of the retina to natural scenes, the very scenes that evolution used to sculpt the function of the retina. Okay? So we're going to replace this simple model with a much more complicated model that takes into account the fact that there are hidden layers or hidden neurons in the retina. So we're just going to assume a convolutional neural network model where you have these two convolutional layers, and then each ganglion cell pools from this convolutional layer with all-to-all connectivity. And then we're going to get a predicted firing rate from any natural scene stimulus. And we're going to work in collaboration with Steve Backus's lab at Stanford, where he can record the response of retinal ganglion cells to many, many natural scenes or movies. Okay? So we're going to train the neural network to mimic the input-output map of a biological retina, in this case from a salamander. Okay? There's many reasons to think this would be a terrible idea. Right? A, it wouldn't work because we wouldn't have enough data. We only have 1 hour's worth of recordings and about 20 cells. B, even if we got it to work, the innards of this model would not at all look like the retina. C, even if we got it to work, the algorithms that are used would be completely different from the algorithms that the retina uses, maybe. Okay? It turns out all of those fears turned out to be, luckily for us, unfounded. So first it worked. Okay? So here's the gray is a trace of a firing of a single salamander ganglion cell over time. All right? The orange is an LN model, and as you can see, it gets a lot of stuff wrong. Okay? Here's the CNN model, and as you can see, it does better than the LN model, and you can quantify this in terms of the correlation coefficient between the model and the actual biological circuit. Now, there's an upper bound on how high this correlation coefficient can possibly be due to intrinsic stochasticity in the retinal response. If you show the same stimulus, the retina doesn't respond exactly the same each time. So that stochasticity sets an upper bound, and our correlation between the model and the retina comes close to the upper bound set by the correlation between the retina with itself. Okay? It comes close to that upper bound. The other models don't come as close. This is an LN model, and this is a GLM model. Okay, so it works, but can we learn anything from this? All right? Okay, so then we can look inside the hidden units of this model and ask, do the hidden units of our model retina look like the hidden units of the biological retina? Okay. And it turns out that's the case. So in harder experiments, you can actually record from the innards of the retina, say from the bipolar cells. We know a lot about the receptive field structure, and it turns out, so these bipolar cells can well be modeled by a linear filter and a nonlinearity. And it turns out the spatiotemporal filters of our model look like that of a bipolar cell. In fact, we can take a second retina and record from a particular bipolar cell in that second retina, then go back to our model and find the best matching internal unit in our model, and we can superimpose the traces on each other. So the purple is our model, our best matching model unit, and the black is an actual recording from a second retina, and as you can see, there's a nice fit. So the interneurons have similar response properties. The hidden neurons in the model have similar response properties to the hidden neurons in the actual retina. So that's good. Okay. Does the thing generalize to decades of experiments done on the retina? Okay. So before we could get veridical models of the retina to natural scenes, experimentalists used their intuition to come up with very interesting artificially structured stimuli that exposed striking retinal computations that couldn't be explained by linear and nonlinear models. Okay? And these stimuli consisted of things like moving bars, moving dots, full field flashes, gratings, and so on. And there's a host of phenomena that have been uncovered about the retina over decades of physiology. Now, we only fit our model to natural scenes. We didn't fit our model to these 30 to 40 years of physiology. But then this actually raises a question. Are those 30 to 40 years of physiology using artificial stimuli at all relevant to understanding the operation of the retina under natural movies? I.e., are the retinal computations elicited by such artificial stimuli at all relevant to understanding retinal computations elicited by natural stimuli? So intriguingly, our CNNs trained only on natural scenes, they actually reproduce these 30 years of experiments. Okay, despite the fact that these stimuli have very different statistics than the natural movies. So in machine learning, you would call that a domain adaptation or a cross-task generalization or something like that. Interestingly, CNNs trained only on white noise do not reproduce any of these phenomena. So natural scenes trigger a whole bunch of nonlinear computations of the retina that are also triggered by these artificial stimuli, but white noise does not. Okay? So now what are some of these phenomena? Why are we so excited about them? These phenomena are kind of striking, and so I'll just give you 3 of the examples. So one is the emitted stimulus response. If you show full field flashes to a retina, okay, of course the retina will respond to each flash. Okay, and there'll be a certain delay. So this flash corresponds to this one, this last flash corresponds to this one. But then if you emit a flash here, the retina will fire a whole lot more. It's as if the retina has learned that for the time being, the world is periodic. And if the world violates that periodicity rule, it'll complain a lot. Moreover, it'll fire at exactly the right time for a range of frequencies within some range. So it not only learns that the world is periodic, it learns the period or the frequency of that periodic signal over a certain range of about 5 to 12 hertz. Okay? Here's another response, which is motion reversal. If a bar moves and then it suddenly reverses, a whole bunch of ganglion cells near the vicinity of that reversal will all complain a lot. So it's as if the retina knows about Newton's first law of motion. Objects that are in motion tend to remain in motion unless they suddenly reverse and then there's some violation of that law. Okay? Another interesting thing is our eyes move around all the time, which means our retinal image is moving around on our retina, but we don't think that the world is moving around, right? The beginnings of that problem is solved in the retina. So there are object motion sensitive cells where if you move the entire scene on the retina, the cell will not fire. But if you differentially move the scene in the receptive field of the cell and move the scene in the opposite direction outside of that receptive field like this, then the cell will fire a lot. Okay? So these are all kind of signatures of the retina acting as a predictive world model for very simple predictions, right? Periodicity, straightforward motion, or the fact that global motion is highly likely, and subtracting out those predictions or signaling violations of those predictions. So it's kind of, so our deep learning model actually gets all of this right. I won't go through all the details, but for example, here's the motion reversal, here's the data, here's our model, okay? Here's, where's the emitted stimulus? Yeah, here's the data, here's the model. So it gets all of these right. So now the question is, let me just go to here. Okay, so now the question is, how is our model getting all of this stuff right? Right? Like, is it enough to just have a good model that explains what's going on, or do we want more? Yeah.
Where's the memory in this model?
Oh, yeah, sorry. Okay. The first layer neurons have spatiotemporal filters. So all of the memory is in the first layer neurons. We trained the temporal filters. Yeah, the spatiotemporal filters were trained on the natural scenes. Yeah, yeah. So, but here's the basic question, right? So, can we understand this model and how it simultaneously accounts for all of the experiments? And interestingly, these are all qualitative phenomena, right? There's qualitative phenomena, this burst, firing here but not firing here. So interestingly, to obtain conceptual understanding, it may be the case that once we have become quantitative and we have a good model, we have to step back and become qualitative to obtain conceptual understanding. So to get there, we need systematic approaches for doing model reduction, I think, on complex deep neural networks. Okay, so that's what we tried to do. Yeah, yeah.
What percentage of retinal ganglion cell
this is very beautiful, but there are multiple classes of RGCs that abstract different features.
Yeah. So
Can you get all of them? I don't know how many there are in Salomon.
so the correlation coefficients that I showed you were an average over all cell types. The variance across cell types was much smaller than the mean average. And there are some cell types that are much more like the distribution of cell types is uneven. There are some cell types that are much more numerous than others. And those ones we get a little bit better because they contribute more to our training data. Yeah, Ara? Yeah, we controlled for luminance and contrast variations to some extent, but not fully. And I don't have a good answer for you as to why we didn't need explicit gain control. Partially, but of course, for any given natural scene, there's huge variations in contrast across the image, and we don't, of course, don't control for that. Yeah. Okay, so here's the basic idea. We kind of, if this is the actual retina and this is, yeah, okay, let me skip the philosophy. Let's focus on this row, all right? So here's our complicated model. So basically what we're going to do is we're going to come up with a method for isolating the interneurons that are important, the hidden neurons that are important for any specific stimulus in generating the ganglion cell response, and we're going to cut out the rest of the circuit. And so for each stimulus, we're going to carve out a subcircuit that's important for that stimulus. Okay, how do we carve that out? So again, we've done it in this paper that will appear at NeurIPS for these 4 stimuli. For this talk, I'll just focus on the emitted stimulus response because that's an easy one to understand. Okay. So the way that we're going to do it is we're going to use methods of attribution for machine learning. So oftentimes when you simplify a model, like let's say you have a complex nonlinear function over a stimulus space, right? What you might want to do is you might want to Taylor expand at the origin and then approximate the function by its expansion coefficients at the origin. And so this Taylor expansion is roughly the receptive field, the space-time receptive field of the neuron. Okay, the space-time receptive field is something that has dominated the way that we think about neurons in sensory neuroscience. But we can use a slightly more sophisticated idea, which is instead of Taylor expanding at the origin, just follow the integral. Okay? So for example, let's say I'd like to explain the height of this function at a particular stimulus. Of course, this is a multidimensional domain, but what I could do is I could just integrate along a curve from the origin to that particular stimulus. So let's say S of alpha is that curve where alpha goes from 0 to 1. I can just integrate the derivative and I'll get the right answer, the exact answer, right? But what I get is I get a decomposition of that integral into each of the multidimensional components. So this is, of course, all I've done is I've taken the gradient of f with respect to s, and then the tangent vector of the curve, taken the dot product and integrated. I will exactly get f of s, okay? But I can decompose the sum into each of the individual components and then attribute the final response to each of the individual components. In the application to the retina, this could be, each individual component could be an interneuron, or it could be a space-time pixel in the image. Okay? So the way we're going to do model reduction is we're going to attribute the response of a ganglion cell to the first layer interneurons to get exactly how much each interneuron contributes. And then each first layer interneuron is well described by an LN model. So by composing those two, we get a reduced model. Okay? Let me just show you, so here, for example, for the emitted stimulus response, we'd like to understand just 2 qualitative things. What mechanism causes a large amplitude burst? And how is the latency of the peak proportional to the period of the flashes? How does it learn the period? Where's the memory, as Michael asked? Okay, so when we apply our attribution methods, which are algorithmic, we find that there are 3 convolutional channels that are important. We can, of course, average over space because the stimulus is spatially invariant. For more complex stimuli, we do dimensionality reduction rather than just spatial averaging. But in any case, we find that 3 filters are important. 1 filter is important for generating the response to each burst. The other two filters are important for generating the emitted stimulus response. So it turns out filters 2 and 6 are important. And it's relatively easy to understand. So what happens is filter 2 has a very fast response, but with a very long memory tail. Okay? And cell type 3 or filter 3 has a slow response with a very short memory tail. Okay? So then what happens is, as you increase the frequency of the flashes, the memory filter will integrate more, and because it responds faster, it'll push the emitted stimulus response to an earlier time. And that's the basic mechanism. Now you could ask, is this consistent with previous works? Okay. So interestingly, there were two major previous models of the emitted stimulus response. One involved an optical resonator circuit. So they looked for, they thought they needed a resonator to learn the periodicity of the signal. They looked for resonant responses in bipolar cell synapses, and they didn't find them. Okay, here's another one that gets the burst, but it doesn't get its latency right. This is the first model that's consistent with all of the data. Now you could ask, what did we do? How did it work out for these other three things? I won't go into the details, but for these other things, when we did model reduction on our deep neural network, we got a reduced model that was previously written down in the scientific literature and had further experimental tests that verified that model. So in some sense, the deep retina had hiding within it all sorts of previously written down models that were tested in further experiments. Okay, so let me, okay, so yeah, so in some sense, this is kind of, it might suggest a pathway for reversing the way that we do science, right? Whenever we're faced with a complex system, at least in neuroscience, we've tried to design or quote unquote intelligently design stimuli to probe what we think is the function of that system. And then for each stimulus, we design a stimulus-specific model for that stimulus. And then some of the models might be inconsistent with each other, who knows? Here, we kind of turn things around. We just throw complicated stimuli at the biological system. We train a complicated neural network. We verify that it works. And then we do model reduction to get an array of interpretable models. And what's nice, at least, is that we find that for all four stimuli, 3 cell types really matter, and we get a unified model for all 4 of these stimuli, which never really existed before. The other cell types are important for other natural scene responses. All right. Okay, so again, it was a lot to take in, but if you're interested, the details are in here. Okay. So let's see, how much time do I have? Okay, 7 minutes. So yeah. Yeah. Sorry, say that again? The first step nature has done for you, but it's hard to do experiments on that. Well, okay, so it used to be hard to train these models. Now we've gotten better at it. Yeah, but we don't have a quantitative description of it. I mean, so my goal is to get quantitative interpretable models that I can simulate on a computer. To say that the brain exists already and so we don't need to simulate it on a computer, I don't understand your objection. Yeah, let's talk afterwards. Huh? Yeah, let's talk about it afterwards, yeah. It'll always be a fun discussion, I know, so. All right, so, okay, okay, let me just, there's a different perspective, so there's a different perspective that we can take on the nature of explanation in both deep learning theory and neuroscience. So here we fit a model to the data, so we required an input-output map collected from experiments from the actual brain, and then we fit a model. Alternatively, can we derive the retina ab initio from first principles under the assumption that maybe the retina is doing optimal encoding of natural movies, right? So this is the other part. So, moving on to the primate retina, which is quite different from the salamander retina, it turns out the primate retina is dominated by kind of 4 major cell types, okay? The jargon is off parasol, off midget, and so on. But these are data from E.J. Chichilinsky's lab on 4 cell types. What are the major features of the 4 cell types? So this is the spatial receptive field, the temporal receptive field, and then the joint receptive field in Fourier space, okay? But what are the, okay, so the question is, why are there so many cell types? What are these cell types good for? In machine learning, just convert cell type to convolutional channel because the retina has a convolutional structure. Okay, this is a major question in neuroscience, right? There's a major experimental effort in neuroscience into cataloging all the cell types in the brain. Yet we have no theory in general for how many cell types we have. And we'd like to try to address this in the retina, at least. So what are the qualitative features of these cell types? So you have the midget cells and the parasol cells. The midget cells have a very small spatial receptive field, but they're slow in time, right? And the parasol cells have a large receptive field, but they're fast in time. Okay, there's a lot of midget cells, but they don't fire very much. And there are relatively fewer parasol cells, and they fire a lot. Okay. So why is there this intertwining between time, space, number, and energy or firing rate? Okay, why does that exist? Okay, so we tried to explain these facts under the following theoretical framework. So the theoretical principle is that the retina may accurately encode natural movies. We need to go to movies and not just natural scenes because we're trying to explain a particular intertwining of time and space. Okay, and we're going to compare a convolutional autoencoder that takes natural movies and just reconstructs them with 1 convolutional channel or 2 convolutional channels. And we're going to optimize the strides and so forth, but holding the overall firing rate and overall neuron number the same in this model and this model. Now, what is the statistics of natural movies that we need? Largely to explain the linear receptive field properties, we only need to think about the second-order statistics of natural movies. They have a very striking second-order structure. So this is the power spectrum of natural movies, both as a function of spatial frequency and temporal frequency. The contours of equal power have this hyperbolic shape, so that there's these 2 tails, one tail at high spatial frequency and low temporal frequency, and another tail at low spatial frequency and high temporal frequency. It turns out that if you try to autoencode natural movies with 1 cell type, for the same number of neurons and same overall firing rate, you do worse with 1 cell type than with 2 cell types. And the optimal 2 cell types that you get specialize to these 2 lobes of the power spectrum. So one lobe goes here, and the other lobe goes here, okay? So if we think about these 2 lobes, this lobe, the receptive field, has high spatial frequency and low temporal frequency. Because the receptive field is small, you need a lot of them to tile space. So you need a lot of them. And because you have a firing rate constraint in our optimization, they must have low firing rates to meet that constraint. On the other hand, so these are the midget cells. On the other hand, you have the parasol cells that have low spatial frequency and high temporal frequency. And because they're low spatial frequency, their receptive field is wide, which means you need a smaller number of them to tile space. And because there isn't that many of them, they can fire more. Okay? That all falls out mathematically and analytically from an optimization. Now, we also simulated a nonlinear autoencoder with rectifying nonlinearities. And this is what we got from our simulation. And this is the data, right? And so you can see that there's a nice qualitative structure where as soon as you add rectification, you also get the on and off types. Okay? All right, so the take-home message is the structure of the primate retina can be derived from first principles, assuming that the retina is acting like an optimal convolutional autoencoder. Okay? And this is in this NeurIPS paper. The final thing I can just state in 1 minute. There are these famous grid cells. This is a single cell that fires at the vertices of a hexagonal grid as a mouse is exploring an open container. A natural question is, could such grid cells arise from a functional constraint? Like what computational task demands that you have grid cells? And we can actually obtain these grid cells by training a recurrent neural network to path integrate, i.e. convert velocity to position. And then, If the firing rates are constrained to be positive, then you get hexagonal grid cells. If they're not positive, you get square grid cells. And we have a theory for why. It turns out positivity induces a three-way interaction between plane waves that generates grid cells, but I can't explain that in the limited time left. But there's a nice mathematical theory for why hexagons emerge in neural networks that path integrate with positive firing rates. All right, thanks.